Today we learn about the topic nitrogen fixation and ammonia assimilation in plants. First we discuss about the properties of nitrogen. That is, the molecular nitrogen is very stable with triple bond. The boiling point of nitrogen. Boiling point of nitrogen minus 195 degree 0.8 degree centigrade. Here is triple bond and also the boiling point minus 155.8 degree centigrade. It is the component, the nitrogen is the component of protein, enzymes, vitamins and alkaloids. That first then we discuss about nitrogen cycle. This is nitrogen cycle. Here the lightning, mainly the lightning fixes N2 to N3. That is NH3 form. Lightning fixes nitrogen to NH3 form. Bacteria like Rhizobium convert N2 to NH3 also. Lightning is the physical matter and some bacteria like Rhizobium also convert N2 to NH3 form. Dead organisms and excreta like urea are decomposed by bacteria into NH3 form and other bacteria into nitrate. These left into the soil for use by plants. Now the nitrogen in atmosphere convert to NH3 form by the presence of nitrogen fixing bacteria. This directly denitrify and again enter into atmosphere. That is known as denitrification. The process is helped by denitrifying bacteria. There is another step. This nitrogen fixing bacteria help in symbiotic form and fix the nitrogen in plants. Animals utilize this nitrogen, the dead organic materials of plants and animals, degraded and again converted to ammonia. That is also converted to nitrate. Then this cycle also complete in another way. There are different ways of nitrogen cycle. Then we discuss about nitrogen fixation. Nitrogen fixation. There are different methods. First, abiological nitrogen fixation. Second, biological nitrogen fixation. First we discuss about abiological nitrogen fixation, that is Herb's process. First, abiological nitrogen fixation. That is also known as Herb's process. Here, synthetic ammonia is produced by passing a mixture of nitrogen and hydrogen through a bed of catalyst, that is iron oxide, at a very high temperature and pressure. In abiological nitrogen fixation method, that is Haber's process, the synthetic ammonia, that is artificial ammonia, is produced by passing a mixture of nitrogen and hydrogen. And hydrogen through a bed of one of the catalyst, iron oxide. Iron oxides. At a very high temperature and pressure. In Haber's process, synthetic ammonia is produced by passing a mixture of nitrogen and hydrogen through a bed of iron oxides, that is the catalyst, at a very high temperature and pressure. Next, biological nitrogen fixation. This is the formula of abiological nitrogen fixation, that is Haber's process. Nitrogen and hydrogen at 500 degrees centigrade and 1000 atmospheric pressure converted to ammonia. Then second, biological nitrogen fixation. It is the reduction of molecular nitrogen to ammonia. The reduction of molecular nitrogen to ammonia by a living cell in presence of enzyme called nitrogenases. The reduction of molecular nitrogen to ammonia by a living cell in presence of an enzyme called nitrogenases. Next topic, some free living organisms also fix nitrogen. First, Clostridium, then Klebsiella, Acetobacter, Rhodospirillum, Anabaena. Clostridium, that is an anaerobic bacteria. It is also a non-photosynthetic form. Next, Klebsiella. Klebsiella, that is a facultative bacteria. Facultative bacteria. That is also a non-photosynthetic form. Third, Acetobacter. It is aerobic bacteria. That is also non-photosynthetic. Fourth, Rhodospirillum. A purple non-sulfur bacteria. Rhodospirillum is a purple non-sulfur bacteria. Purple non-sulfur bacteria. It is a photosynthetic form. Next, Anabaena. It is a cyanobacteria. Also a photosynthetic form. Some microbes become associated with other organisms and fix nitrogen. That is known as symbiosis. Here some examples of symbiosis. First, system and the names of symbionts. First, system lichens. Lichens. The two partners are the cyanobacteria and fungus. Cyanobacteria and fungus. Second, bryophyte. That is the combination of cyanobacteria and anthocerus. Cyanobacteria and anthocerus. Third, pteridophyte. Here also the combination of cyanobacteria and azolla. Cyanobacteria and azolla. Next, gymnosperms. Here also cyanobacteria and cycas. And cycas. Next, angiosperms. Here non-legumes and actinomycetes. Non-legumes and actinomycetes. Next, angiosperms. System angiosperms. There are two types, that is legumes and rhizobium. There are different cases in systems angiosperms. There are mainly three systems. First, the angiosperms with non-legumes and actinomycetes. Second, in case of angiosperms, legumes and rhizobium. Again, in some cases of angiosperms, Brazilian grass and azospirillum. Brazilian grass and azospirillum. Non-legumes, Brazilian grass and legumes. These are different types of angiosperms. Collaborate with actinomycete, rhizobium and azospirillum. Now we discuss about some symbiosis and their examples. Next topic, mechanism of biological fixation of nitrogen. Mechanism of biological fixation. Of nitrogen. This recurs. The requirement for N2 fixation are the requirements. First, molecular N2. Molecular nitrogen. Second, strong reducing agent like NAD or FAD. Strong reducing agents like NAD or FAD. Third requirement, source of ATP, that is adenosine triphosphate, source of energy. That is always ATP. To transfer H2 from NADH2 or FADH2 to dinitrogen. H2 from NADH2 or FADH2 to dinitrogen. The fourth requirement, enzyme nitrogenase, an enzyme nitrogenase. And fifth requirement, compound for trapping toxic ammonia. A compound for trapping toxic ammonia. Molecular nitrogen strong reducing agents like NAD or FAD, source of energy ATP, and say nitrogenase. Then compound for trapping toxic ammonia. The NADH2 and FADH2 and ATP are produced by photosynthesis and respiration. The NADH2 and FADH2 and ATP are provided by photosynthesis and respiration. Then the general equation for N2 fixation equation for nitrogen fixation. N2 plus 16 ATP plus 8 hydrogen ion plus 8 electron convert to 2 NH3 plus 16 ADP, adenosine diphosphate, ATP converted to ADP, plus 16 Pi. This is the equation for general equation for N2 fixation. Next topic, fixation of leg hemoglobin in reduction of N2 to NH3. The function of leg hemoglobin in reduction of N2 to NH3. Function of leg hemoglobin in reduction of N2 to NH3. In root nodules of leguminous plants, the symbiotic nitrogen fixing bacteria Rhizobium resides. The bacteria Rhizobium resides inside the root nodules of leguminous plants. The protein nodulin help for maintaining nodule function. The protein that seen in nodule helps for maintaining nodule functions. One of the protein, nodulin, helps for maintaining nodule functions. The leg hemoglobin is produced as a result of interaction between bacterium and legume roots. The leg hemoglobin is produced as a result of interaction between bacterium, that is Rhizobium, and legume root. Rhizobium gene codes for heme part. The rhizobium gene codes for heme part and legume root cell gene code for globin part. Rhizobium gene codes for heme part and legume root cell gene. Legume root cell gene code for globin part. Both coded products together constitute protein leg hemoglobin. That is heme part and globin part. Together constitute protein leg hemoglobin. It acts as oxygen scavenger. This leg hemoglobin act as oxygen scavenger. So that the enzyme nitrogenase convert N2 to NH3 under anaerobic condition. Then the step of biochemical nitrogen fixation. The host plant cytoplasm, that is example one of the legume pea, and rhizobium in nodule, the bacteria rhizobium in nodule to form leg hemoglobin. This leg hemoglobin enter into TCA cycle. Then here some part of keto acid and ATP are coming out. That also form uracid and other amino acids. The next part, from this TCA cycle, 2 electron and 2 H plus ions are formed. Here the ammonium N2 convert to diamide to hydrazine and ammonia. Then the uracid and other amino acid. The end products are nitrogenase, Fe and FeMO complex. Here also another nitrogenase, iron and FeMO complex plus ammonia. That is the flowchart of the step of biochemical nitrogen fixation. The next portion, nitrate and ammonia assimilation in plants. Next topic, nitrate and ammonia assimilation in plants. Nitrate and ammonia assimilation. We discuss the production, fixation and then the assimilation of ammonia in plants or by plants. The next topic, nitrate and ammonia assimilation by plants. The first step, Nitrate is absorbed by most plants, reduced to ammonia. Nitrate is absorbed by most plants, reduced to ammonia with the help of two different enzymes. There are two different enzymes are helped for this process. Nitrate is absorbed by most plants, reduced to ammonia with the help of two enzymes. The enzymes are nitrate reductase, the nitrate reductase and nitrite reductase. Two enzymes, nitrate reductase and nitrite reductase. Nitrate and nitrite reductase. The conversion of nitrate to nitrite is catalyzed by the enzyme nitrate reductase. That is NO3 minus plus NADH plus hydrogen ion by the help of enzyme nitrate reductase. Convert NO2 minus plus NAD plus water molecule. NO3 minus plus NADH plus H plus in presence of enzyme nitrate reductase. Convert NO2 minus plus NAD plus and H2O molecule. In the second step, this is the first step. Then the second step, nitrite formed is further reduced to ammonia. The nitrite formed is further reduced to ammonia. That is the second step. That is nitrite further reduced to ammonia. It is catalyzed by another enzyme nitrite reductase. The nitrite present in cytosol then transported to chloroplast or plastids, different types of plastids, where it reduced to ammonia. Here the nitrite present in cytosol then transported into chloroplast or plastids, where it reduced to ammonia. We can write the formula. NO2 minus, here is the NO2 minus plus 3 NADPH plus 3 hydrogen by the help of an enzyme nitrate reductase. Nitrite reductase and converted to NH3 ammonia plus 3 NADP plus molecule. The accumulation of ammonia that is toxic to plants. So ammonia can further oxidized to nitrite and nitrate by the microorganism in the soil or water. Here the ammonia is poisonous or toxic to plants. So this ammonia can further oxidized to nitrite and nitrate. The toxic ammonia poison to plants. So toxic to plants. So ammonia oxidized to nitrite and nitrate. Nitrite and nitrate by microorganisms in the soil or water. The next topic, amino acid synthesis by plants. Amino acid synthesis by plants. Next topic, amino acid synthesis by plants. Ammonia is the major source for the production of amino acids. Ammonia is the major source for the production of amino acids. There are different types of amino acids. Amino acids have two important chemical groups. Amino acids have two important chemical groups. First amino group and second carboxyl group. Amino acid have two important chemical groups. The first amino group. Amino group, that is NH2 group. Amino group, that is NH2, and the second carboxyl group. Carboxyl group, that is COOH group. Then the structure of an amino acid. Structure of amino acid. We can draw. R means alkyl group. Alkyl group. Next, reductive amination reaction and transamination reaction. First, reductive amination reaction. Reductive amination reaction. In this reaction, ammonia combined with keto acid. In amination reaction, ammonia combines with keto acid. The important keto acid is alpha keto glutaric acid produced during Krebs cycle. In this reaction, ammonia combined with keto acid. The important keto acid is alpha keto glutaric acid produced during Krebs cycle. The keto acid undergoes enzymatic reductive amination. Keto acid undergoes enzymatic reductive amination to produce an amino acid. Here, alpha keto glutaric acid. Keto glutaric acid combined with NH3 ammonia in presence of an enzyme. To form glutaric amino acid. Glutamate dehydrogenase. Glutamate dehydrogenase. Act as a catalyst. Alpha keto glutaric acid plus NH3 in presence of glutamate dehydrogenase to form glutamic amino acid. This is known as reductive amination reaction. Once more, in amination reaction, ammonia combined with keto acid is alpha keto glutaric acid produced during Krebs cycle. Second, transamination reaction. Transamination reaction. Here, the reaction involves transfer of amino group from already synthesized amino acid to the keto acid. The reduction involves transfer of amino group from already synthesized amino acid to the keto acid. Here, alpha keto glutaric acid. Keto glutaric acid plus aspartic amino acid, one of the amino acid. Aspartic amino acid in presence of an enzyme transaminase. Transaminase to form glutamic amino acid and oxaloacetic acid. To form glutamic amino acid and oxaloacetic acid. Oxaloacetic acid. A number of amino acids are produced by this method. Now we complete this part. If you like this, please like, share and subscribe.